Hello everyone, my name's Dr. Bob Brewin. I am a lecturer in physical geography based at the University of Exeter, and I'm going to be presenting on optical remote sensing for marine applications. So as I say, I'm based at the University of Exeter, and if you'd like to learn some more information about me, please follow the link to my webpage. Okay, so a little bit about myself. Well, I was actually educated at the University of Plymouth. I was a keen surfer when I was younger, and although we didn't learn too much about ocean sciences while I was at high school and during my A-levels, I undertook a undergraduate degree in surf science and technology, motivated by my passion for surfing. And while doing this degree, I learnt more and more about oceanography, and then afterwards I did a MSc in applied marine science, and then did a PhD in marine remote sensing with a focus on ocean colour. I also got some experience as a lecturer and demonstrator while I was at the University of Plymouth. After that, I spent about 9 years at Plymouth Marine Laboratory, where I worked as a research scientist in the remote sensing group there. During this period, I actually did a 2-year ESA postdoctoral fellowship, focusing on looking at phytoplankton community structure using satellite observations. And I also worked as a scientist for the National Centre for Earth Observation. I spent about 6 months of my time over that 9-year period going to sea. I did 4 Atlantic meridional transect cruises down the centre of the Atlantic, collecting oceanographic observations for satellite validation. And since July 2019, I've been based at the University of Exeter at their campus in Penryn, which is right in the southwest tip of the UK. And I've been based there for about 1.5 years. I lecture on modules in biological oceanography and remote sensing. Okay, a little bit about my research interests. I do a lot of work using remote sensing observations, ocean colour particularly. I'm interested in the relationship between ocean colour and phytoplankton in the ocean. So these are these microscopic single-celled algae that are the primary producers of the sea. And there is quite a wide range of diversity of phytoplankton, and a lot of the science I've done is trying to detect different communities of phytoplankton in the ocean and how this data can be useful for understanding better marine carbon cycling processes. As I mentioned in the previous slide, I like to go out into the field a lot. I've spent a bit of time on these Atlantic meridional transects, collecting bio-optical observations for using satellite validation, as well as doing other experiments at the sea. And I also have a bit of a passion for citizen science, so I have projects where we've been using surfers as a platform for collecting ocean observations in the near shore. Also been working with my brother on developing 3D printed water quality and clarity tools. This is a miniature Secchi disc in the bottom right here for use in lake citizen science projects. Okay, so that's a bit about me. Now, what I plan to do in this lecture, which is going to be about 1.5 hours long, I've split it into 3 different parts. The first part, I'm going to sort of give a bit of background on ocean colour and the theory behind ocean colour. And then we're going to focus in on 2 ocean colour applications. Firstly, using ocean colour for understanding carbon cycling in the ocean, and then using ocean colour to look at monitoring climate change in the ocean. So I'll start by looking at the ocean colour theory. So in this part of the lecture, I'm going to go over what is ocean colour, a bit of the history of ocean colour. We're going to go a little bit into the detail of optical properties of water and its constituents, how we measure ocean colour in situ, so that's in the ocean or on the ocean, and also finishing by looking at satellite remote sensing of ocean colour. So sort of basically to give you guys the basics a basic foundation from which we can then go into some of the applications in the later parts of the lecture. Okay, so what is ocean colour? Well, ocean colour is defined as spectral variations in visible light reflected from water. So basically, sunlight enters into the water, it's absorbed, scattered and reflected by water itself, but also the constituents that make up the water. The particles in the water, the dissolved substances that are coloured. Now, these processes of absorption, scattering and reflection vary with wavelength of light. Typically, what we do in terms of satellite remote sensing is use visible radometers on satellites to look at ocean colour, but we can also look at ocean colour with our own eyes, which are tuned to the visible portion of the electromagnetic spectrum, and other sensors like cameras and radometers. Okay, so starting from the beginning, a little bit of the history. Well, ocean colour has long inspired poets and artists. You can just see by some of the beautiful pictures by artists here who've had a passion for ocean colour. Now, ocean colour was also used in early navigation. It was used for looking at bottom topography, used for predicting the presence of icebergs, river discharge. There are observations made by some of the early navigators on the occurrence of fish and seabirds in relation to the colour of the ocean. And there's some nice examples, for instance, from Hudson's first voyage in 1607 and William Scoresby in 1820. Some of these observations made by these navigators, first observations came from these sources. Now, if you'd like to learn a little bit more about the history of ocean colour and ocean optics, there's this wonderful book by the late Marcel Vernard. If you can't access this book, I haven't actually been able to get a copy of this yet, though a colleague has one. Marcel did publish his PhD thesis, from which a lot of this book, the work in this book, is based on in a ResearchGate. So if you follow that link at the bottom, it will take you to a copy of his thesis, and there's lots of really interesting information about the history behind ocean colour. Now, in terms of our further understanding of what does the ocean colour mean, it's quite clear that the colour of the ocean can change quite considerably with time and place. And it has made scientists, certainly some of the early scientists working on ocean observations, think what's responsible for these changes? Can we quantify the responsible agents that are controlling the colour of the ocean? Why should we care about the colour of the ocean and some of these agents? And this is just a lovely image taken from a CEOS report in 2018, which just illustrates the diversity of colours you can see in the ocean. Okay, but in terms of quantitative physical remote sensing, it was really the work by Raman in 1922 who first showed that the colour of pure water is blue. And I've just took this extract from the work of Marcel Vernard, actually, and some of those earlier links that he found from Raman. And it says, a sufficient deep layer of pure water exhibits bimolecular scattering, a deep blue colour, more saturated than skylight and comparable in intensity. The colour is primarily due to diffraction, the absorption only making it a fuller hue. The theories hitherto advanced that the dark blue of the deep ocean is reflected skylight, or that it's due to suspended matter, are discussed and shown to be erroneous. So the work by Raman basically showed that pure water has this colour, this reflected colour of blue. Now, why is that? Well, if you look at the plot on the right here, we have the absorption coefficient on the y-axis on the left and the backscattering coefficient on the y-axis on the right. Backscattering shown in dotted line and absorption in the solid line. And what you can see is pure water absorbs strongly at red wavelengths, down at 700, 600 nanometers. And this decreases a little bit in the green wavelengths, but it's at its minima in the blue wavelengths. Alternatively, backscattering by pure water is highest at blue wavelengths and decreases exponentially with as a power function with wavelength. Now, the reflected lights can be defined as a ratio of upwelling irradiance to downwelling irradiance, shown here on the equation on the left. And this is roughly proportional to the backscattering coefficient divided by the absorption coefficient. So, if pure water absorbs a lot in the blue and reflects highly, or backscatters highest in the blue, we would expect to see a very strong blue reflectance and a lower reflectance as you go into the green and the red wavelengths, where the absorption increases and the backscattering decreases. And this is just an example of that. This is an in-situ observation of ocean colour made out in the North Atlantic, some of the bluest waters in the ocean, right out in the North Atlantic oligotrophic gyre. And you can see the reflectance on the y-axis here, which is actually on a log scale as a function of wavelength, peaking at blue wavelengths within around 400 nanometers and decreasing with increasing wavelength. So it's well known that the colour of the ocean is blue. But of course, when we look at the ocean, we see many different shades of colour. So why is that? Why is the ocean not blue everywhere? Well, it's from the simple fact that the ocean contains other constituents beyond pure water. There's particles in the ocean, there's dissolved substances. So this sort of just goes through a similar flow here, but starting on the left here we have our pure seawater example, where pure seawater dominates the reflective signal. There's nothing really else in the water, and such that the waters look very blue. But then if we increase the amount of particles in the ocean, if we put lots of phytoplankton in the ocean, now phytoplankton contain pigments like chlorophyll, which absorbs strongly in the blue, less in the green, and a little bit in the red. Actually, phytoplankton have evolved to absorb strongly in the blue wavelengths because pure seawater doesn't. Basically, they can harness the most light at that wavelength. Now, if we fill the ocean with phytoplankton and with chlorophyll pigment, we would start to see a greening of the ocean, shown in the image at the bottom. Similarly, if we go into coastal environments where there's a lot of sediment, that sediment absorbs and scatters light in such a way that the oceans look quite bright brown. Alternatively, if we fill the oceans with colour dissolved organic matter, perhaps from rivers flowing off the land as an example, we would now colour dissolved organic matter absorbs very strongly in the blue. Not thought to backscatter light. We start to see these sort of very dark brown waters. So by looking at the colour of the ocean, we can begin to understand what are the constituents in the water and what are their concentrations. Okay, so a few useful definitions to get your head round. In terms of ocean optics, there's two types of optical properties. There's apparent optical properties. Now, these are dependent on the inherent optical properties, which I'll talk about in a moment. So not only on the optical properties of the particles or the constituents or the dissolved substances, but also dependent on the directional distribution of the light field in the sea. So thinking about where the sun is in the sky, how that light enters the ocean at certain angles and intensities. And these apparent optical properties are commonly used are the water leaving radiance, reflectance, and the diffuse attenuation coefficient of seawater. So these are the optical properties, for instance, some of them that you would see from satellites, certainly the water leaving radiance. So in order to get at the inherent optical properties, one needs to understand the directional distribution of the light field. Now, inherent optical properties are the optical properties of the water and its constituents, and they are independent of the directional distribution of the light field in the sea. So this is the absorption, the backscattering coefficients, and also other optical properties like the beam attenuation coefficient. Now, the inherent optical properties can be considered as specific or bulk. Now, specific inherent optical properties are the individual scattering and absorption components per unit concentration, and they're often indexed by this superscript star here, where the biomass specific absorption coefficient of phytoplankton, denoted A star B, is the absorption coefficient of phytoplankton divided by its concentration. Now, the bulk inherent optical properties, however, are the individual scattering and absorption components per unit concentration multiplied by the concentration. And so these will vary depending on the specific IOPs and the concentration, but they give us a route into using optical observations to get at the concentrations that are important for biogeochemistry and other applications. Okay, so if we go to our bulk inherent optical properties, our absorption coefficient and our backscattering coefficient, they can be decomposed into various components that influence them. There's the pure water itself, which we saw in the slide earlier. There's the absorption and the backscattering coefficients of phytoplankton and detritus and colour dissolved organic matter. Now note that some of these group, so for instance the absorption coefficient of detrital and CDOM absorption is often considered as one because the spectral slopes of these absorption coefficients are very similar. Similarly, it's difficult to partition out the effect of phytoplankton and detritus on the particulate backscattering coefficient shown in the bottom right there. Now, in coastal and optically complex waters, often many of these constituents do not covary. So, for instance, the phytoplankton biomass in the water does not necessarily covary in a predictable manner with the detrital pool and with the colour dissolved organic matter pool. And these types of conditions are often referred to as case 2, optically complex waters. We also have a case in the ocean that have been referred to as case 1 waters. Now these are typically your open ocean conditions that are normally, but not always, far away from the coastline. And in these cases, the phytoplankton biomass, so the chlorophyll concentration in phytoplankton, are found to covary in a predictable manner with the other pools in the ocean of detritus and CDOM, and meaning that the inherent optical properties can be tied with reasonable confidence to the chlorophyll concentration. So in a way, it allows us to treat, we have to consider the algorithms we use that we apply to ocean colour to derive chlorophyll concentration differently depending whether we're in this case 1 or these case 2 environments. Now, having broken our bulk absorption coefficient down into its different components, we can then use those components to get information about the environment. So the phytoplankton coefficient, denoted AB here, can be used to get the total chlorophyll pigment, the photosynthetic pigment in all of phytoplankton. It can also look at pigment composition, so other pigments potentially. You can get information on the size of the phytoplankton, the community of phytoplankton, potentially even the amount of carbon and the attenuation by the different phytoplankton communities. The ADG, so the detrital and sedum pool, can tell us information about the sinking of material, about coastal erosion, river deposition, dust, volcanic activity, recycling and light attenuation. And the sedum coefficient we can use to get information about dissolved organic carbon, which I'll be talking about a bit later, proportions of humic and fluvic acids, semi-liable and refractory fractions, and the photodegradation status of the sedum. So optical properties give us a route into understanding these different components that make up the water, and as a result allows us to use this information to look at scientific questions. The backscattering coefficient by particles can be used to look at suspended particulate matter in the ocean. Light attenuation can also be used to look at phytoplankton size structure and even potentially phytoplankton carbon. And it's been used to monitor particular organic carbon and particular inorganic carbon. And we'll be talking about these a bit later when we look at the applications in the context of the carbon cycle. Now, some of the first in situ measurements of ocean colour were done using tools known as the Secchi disc, which is a simple white disc that you lower into the ocean and measure the depth at which it disappears from sight, and this tells you something about the clarity of the ocean. And another tool called the Frel-Ulé colour scale, and here you typically use your Secchi disc at half the Secchi depth, so a bit shallower, and then you look at the colour of the disc in relation to a colour scale. And these measurements have been used since the late 19th century and used to map the colour of the oceans, and actually with remarkable accuracy. So if we look at this map in the middle here of the colour of the Atlantic Ocean from measurements collected using the Frel-Ulé colour scale, and we compare that with a satellite image of chlorophyll concentration, we can see how these discrete observations of colour made at the start of the 20th century reflect remarkably well against the satellite observations. You can see the oligotrophic gyres in the North and South Atlantic, these blue regions in both the images. You can see the equatorial upwelling regions and the higher chlorophyll and higher and greener waters in the polar regions. Now, another in situ measurement of colour that's used commonly since the 1930s has been measurements collected using the continuous plankton recorder. So this is a device towed on the back of ships of opportunity that collects and filters the ocean and collects phytoplankton, zooplankton, and other particles on the filter pads, and then these are looked at and compared to a colour scale of greenness. And these observations, as I say, date back to the 1930s, and you have this phytoplankton colour index that they take from these observations, that they look at changes in phytoplankton biomass over long time periods. Now, in terms of more modern observations of ocean colour, we typically use radometers. So this is an example of some above water radometry that we did on a cruise in the Atlantic, on the Atlantic Meridional Transect, as part of the ESA funded AMT for Sentinel FRM project. And we use these radometers to look at the colour coming out of the ocean, to measure the downwelling irradiance, and also to correct for sky reflectance and get remote sensing reflectance observations, which is what the satellite also sees. And we can use those for validation. We can also do in-water radometry, where we put these radometers into the ocean, we profile with them, or we put them on floating devices, and we measure the colours of the ocean, the downwelling and upwelling irradiance just below the surface. And we can also take in-water measurements of inherent optical properties by pushing absorption and backscattering meters on profilers, or even monitoring in underway mode. So that's as the ship moves, it pumps water surface water into the scientific laboratories, and we can set up backscattering and absorption meters to measure the IOPs continuously as the ship moves. Now, satellite measurements of ocean colour. So this is a beautiful image actually taken from the European Space Agency MERIS sensor on the Envisat platform. And this was back around December, I think 2011, 2012, and it's somewhere in the Southern Ocean, a little bit sort of east of the Falkland Islands, and it shows a beautiful coccolithophore bloom, which is those bright turquoise waters you see in the middle, merging with what we believe is probably a large diatom bloom, which is where you see those dark greens. Diatoms can have very high chlorophyll A concentrations, which show this greening of the water. Now this just illustrates how powerful satellite observations can be for collecting observations over large scales. Now, satellite remote sensing of ocean colour has been considered among the biggest achievements in biological oceanography in the 20th century. And why is that? Well, it's revealed oceanography's chronic problem of undersampling. The fact is, you take out a ship into an environment like that on the right here, where you have all of this dynamic physical, biophysical processes happening. How can you in any way understand that environment from single discrete observations on a ship? And yet satellite observations give us this synoptic view. It's shown the dominance of mesoscale processes in the ocean in determining the spatial distribution of phytoplankton, shown the effect of topography on phytoplankton biomass, the complexity of the seasonal progression of phytoplankton blooms, and the magnitude of the interannual variability. Now, some of the first observations of using ocean colour in the context of phytoplankton and chlorophyll A detection was by work in the 1970s and 80s by Hovis and Gordon and others, and they used the Coastal Zone Colour Scanner, which was a proof of concept mission launched by NASA at the time, shown in the top left here. And they showed that these observations of colour from the Coastal Zone scanner correlate very well with the pigment concentration, as shown in the image on the right here, from that transect along the edge of Florida in this case, with the observations from the ship overlaid on those from the satellite. Now, ocean colour, since the launch of the colour zone, the Coastal Zone Colour Scanner in the 1970s, which was the first proof of concept ocean colour mission, there was a bit of a gap until sort of the late 90s. Since then, there has been a suite of satellite sensors that have been launched in space that monitor the colour of the ocean, as listed here. Now, some of the basics with satellite ocean colour observations is it's first worth noting that as the satellite orbits the Earth and looks down at the colour of the ocean, around 90% of the signal that the satellite's seeing is coming from the atmosphere, only around 10% from the ocean, and that's on a clear sky image. Of course, the problem with ocean colour is we can only really see during clear skies, so when it's cloudy we can't see the ocean. But in the cases where we can see the ocean, we need to do careful atmospheric correction to remove the signal from the atmosphere, as shown here. And once we've removed that atmospheric signal, we can look at that 10% of the water leaving, 10% of the signal coming from the ocean, and then use that to start looking at the optical properties of the water and the constituents within it, as we've seen it. The other thing to bear in mind is that ocean colour is only representative of the first penetration depth in the ocean, and that varies depending on the optical constituents in the surface, but basically means that we can't see below around 40 to 50 metres max, and that's in the clearest ocean. Often the signal is much shallower than that, in the order of, or representative of the sort of top 10 metres of the open ocean. Now, once we've corrected for our atmosphere and we've got our ocean colour observations, we can then relate the colour of the ocean to the properties we're interested in. Now, this is typically done using empirical algorithms such as that shown at the left, which basically uses a ratio of blue to green light and relate that to the chlorophyll concentration, the principal pigment in phytoplankton responsible for its biomass. Or we can use more semi-analytical algorithms where we take our reflectance and break it down into our inherent optical properties, our absorption and backscattering, which are then further broken down into the different components that make them up. And then if we use specific inherent optical properties, we can start trying to estimate their concentrations. Now, this just illustrates a data set I'll be talking about in a little bit from the European Space Agency Climate Change Initiative, which is a merged ocean colour product. And these are maps of the chlorophyll concentration from that product, varying with season. So it's going through each month here over the entire time series, which is around 22 years now, though the images on the left was produced a while ago, so we don't have the data right up to 2020. And as I'll be talking about a bit later on, fluctuations in ocean colour have also been found to be tightly linked to climate variability. Okay, so that's part one out the way. We now know what ocean colour is. You've learned a bit about the history of ocean colour, the optical properties of the water, and the constituents that make up the water. We've learned a little bit about some traditional in-situ measurements and some more modern in-situ measurements of ocean colour, and we've introduced the concept of satellite remote sensing of ocean colour. Thanks for listening.